Hello, everybody. Welcome to Ms. Maguire online video lecture. And today we're covering a cellular level of organization, cells, the living units. And we're going to cover today the physiology part. Okay, so let's go ahead and begin. So we will talk today, we're going to start this lecture talking about stem cells. Now, a stem cell is an unspecialized cell that can divide without limit as needed and can, under specific conditions, differentiate into specialized cells. And in addition, stem cells have unique abilities to self-renew. So stem cells, if you want to define them, we would say that they have ability to self-renew and differentiate into different cells to create functional tissues. Right, so what I want to say over here that actually, pick up my pen. So for example, if you have a cell, so here if you have a cell, and if this cell divides, right, so it's kind of splits into half, right? So we have a cell division, this binary fission, or whatever you call it. So if this cell divides into two cells, you don't have your parent cell anymore, right? Now, stem cells are different. If stem cell divides, right, it's gonna create two cells, right? When cells divide, it make two cells, but one cell became a stem cell again. So that's the ability to self-renew. And because we now have, so we used one stem cell, right? But we have another stem cell. We pretty much can create and differentiate more cells, right? That makes sense. Okay, now stem cells are classified into several categories. So the first is totipotent embryonic stem cells. Now they can differentiate into all embryonic and adult lineage of cells. Then we have pluripotent embryonic stem cells. They can give rise to all cells in an adult. So when these totipotent cells differentiate into endoderm, mesoderm, and ectoderm line, now those pluripotent cells, ectoderm line cannot differentiate into mesoderm, and mesoderm cannot differentiate into ectoderm anymore, right? But they still will give rise to all the types of cells in a human body. Then we're moving to multipotent stem cells. They can differentiate into multiple cells within a lineage, right? So you can see the multipotent cells here that differentiate into lung cells, on pancreatic cells, into muscles, or red blood cells, or skins and neurons. After multipotent, we have oligopotent cells that can differentiate into one or a few different cells only. In unipotent, they are fully specialized and can only reproduce to generate more of its own specific cell type. So you can see the potency of cells decreasing here. So we start with the cells that can differentiate into any type of cells. Then cells that differentiate only within their lineage, or cells that differentiate into multiple cells. And finally, we get to the cells that can only produce its own kind, right? So this is how cells are formed. Now, if you wonder why we even talk about stem cells, because this chapter, cells are living units, we're covering cells. We started with the cell theory that tell us that cell is the smallest functional and structural unit of life, and all cells come from pre-existing cells. Now, if cells come from pre-existing cells, then we must have these stem cells that are undifferentiated. They will differentiate into different lineages and different group of cells that produce all functional tissues and organs of living organism, right? Now here, for example, we can look at hematopoiesis, that is differentiation of multipotent stem cells into blood cells. And in this example, we start with multipotent hematopoietic stem cells that gonna differentiate into common myeloid or common lymphoid progenitor cells. Now, those progenitor cells then differentiate into megakaryocyte that will give rise to platelets, erythrocyte, mast cells, or myeloblast that will differentiate into different type of white blood cells, basophils, neutrophils, eosinophils, and monocytes that can further differentiate into macrophage or dendritic cells. And common lymphoid progenitor will give rise of natural killer cells or small lymphocytes that then will be differentiated into T lymphocytes or B lymphocytes. The B lymphocytes will differentiate into plasma cells to produce antibodies, right? So now, if we go back, we can see that we have our multipotent cells. We start with multipotent cells, and then we will get to oligopotent or unipotent cells, or very often we just call them now progenitor cells instead of stem cells. But sometimes we use stem cells anyway. Now, not only cells are formed, they differentiate, right, to create all organs and tissue. Cells also sometimes need to die. Now, there are several controlled processes that allow your body to get rid of old, damaged, non-functional cells. And one of these processes is autophagy. Autophagy means self-eating. So cells that become obsolete or damaged need to be taken out of system. Autophagy is the process of disposing of non-functional organelles and cytoplasmic bits by forming autophagosome, which can then be degraded by lysosome. So in example, let's say we have mitochondria that is damaged, not working. So the autophagosome will be created, and it will be created, this vesicle created from endoplasmic reticulum. Then it will fuse with lysosome, and lysosome will digest this non-functional organelle. So that's autophagy. Another control cell death is apoptosis. It's a programmed cell death causes certain cells, for example, cancer cells, infected cells, or old cells, to neatly self-destruct. Now, apoptosis actually begins with mitochondria. So mitochondria is directly involved in the process of apoptosis. Very often on my exam, I have this question, what organelle is essential for apoptosis? And many students who are not watching my videos, they answer it's a lysosome. Because, yeah, you're kind of thinking about, yeah, lysosome is digest everything, break down stuff, so it should be lysosome. And yeah, lysosomes are important in the process of autophagy over here, but apoptosis begins with mitochondrial membrane leaking chemicals that activate enzymes called caspases. So mitochondria first leaks chemicals. Caspases are activated. Now, caspases cause degradation of DNA and cytoskeleton, which leads to cell death. Dead cell shrinks and phagocytized by macrophages. So let's compare apoptosis and autophagy. But first, autophagy and apoptosis can be induced and regulate cell death independently. So they induce concomitantly. So that means their function will pretty much be independent, right? So over here on example A, now cell can destroy itself either through autophagy or apoptosis. However, autophagy and apoptosis can lead to one another. One leads to the other. So here, for example, autophagy leads to apoptosis, or when we start process of apoptosis, we digesting organelles inside as well, so that can lead to cell death. Right? One leads to another. Or actually, autophagy can antagonize apoptotic cell death by preventing the accumulation of damaged DNA and ER stress products. So, for example, you have cell, right? And you have damaged DNA. And if you have damaged DNA, the cell want to destroy itself through apoptosis. However, if autophagy helps to get rid of this damaged DNA or non-functional organelle, then we don't need apoptosis to happen and cell will survive. It just pretty much removed the damaged part, non-functional part, so it doesn't need to die. Right? So that's apoptosis and autophagy. And we're gonna here, we will compare also apoptosis, autophagy, and necrosis. Compare it to a normal cell. So this is electron microscope photographs of a normal cell. You see how apoptotic cells look nice and neat here. Autophagy, you can see a lot of different processes going inside, right? So cell pretty much creates a lot of this vesicles inside with the damaged organelles or some cytoplasmic debris and destroying them. And you see how different necrosis looks over here. It's not a neat process. The cell is swelling, and necrosis always will lead to inflammation when apoptosis and autophagy do not lead to inflammation. So you can look at some features like general morphology. Apoptosis cells shrink, in necrosis swell. Chromatin condensation is intensive in apoptosis, so chromosome condenses. It condenses slightly when we have autophagy and no condensation during necrosis. Caspase activation, this is what is activated by mitochondrial chemicals, enzymes. We have activation of caspases in apoptosis only, no activation in autophagy or necrosis. Right, and another important information I already told you to remember: necrosis causes inflammation, and it's not programmed death. It's usually because of some tissue damage, some inflammatory processes that cells die, even perfectly normal cells that's supposed to function, but they just affected by either your bacteria damaging them, or it can be just mechanical trauma. Oops. Okay, another part of this lecture is transport. We know that membrane transport is a movement of chemicals, molecules, and ions across a cell membrane. We have two types of transport: active transport and, oh, I'm sorry, that's a passive transport and active transport. Now, there are several differences between active and passive transport. First, that passive transport the substances, they move down their concentration gradient. So they move from high concentration to low concentration. In active transport, substances move against their concentration gradient. So they move from low concentration to high concentration. Active transport requires ATP, and passive transport does not require ATP. Okay, so let's look at examples of passive transport. So here's our extracellular fluid and cytoplasm inside the cell and phospholipid bilayer that makes cell membrane that separates extracellular and intracellular fluids. So first type of passive transport is diffusion, when small non-polar molecules passes through the phospholipid bilayer. We have another type of diffusion called facilitated diffusion. So we move from high concentration to low concentration. ATP is not used, but we move through non-specific channels, right? So channels facilitate the diffusion. Or it can be facilitated diffusion through a specific channel. So again, we move from high concentration to low concentration. No ATP is needed. But if you look here, all different type of molecules move through a non-specific channel. And this is the specific channel for one particular chemical. For example, it can be potassium or sodium ions. If it's a channel for potassium ions, then sodium ions cannot move through it. If it's for sodium ions, then potassium cannot move through it. Calcium, chloride, they cannot move through this specific channel. They need their own specific channel, right? So facilitated diffusion through a permis specific for one chemical, binding of substrate causes change in the channel protein, right? And osmosis. So osmosis is also a diffusion, but it's a diffusion of water through a specific water channel protein or through the phospholipid bilayer. So either water can move through kind of like simple diffusion through phospholipid bilayer, or it will move through specific water channel called aquaporin. But in in all these four examples, we're moving from high concentration to low concentration, and we do not use ATP. Now, active transport, we will move against the concentration gradient from low concentration to high concentration, and ATP molecule will be used. Active transport is divided into primary and secondary active transport. So example of primary active transport is sodium-potassium pump. So it's shown right here. Shown, let me see. Yeah, right here. So that's a sodium-potassium pump. So again, we have extracellular fluid, we have cytoplasm. We have a special protein that is called sodium-potassium pump. We use ATP energy, and we move sodium out and potassium in, right? So it's not an equal exchange. So every time we move 3 sodium out and 2 potassium in, using, again, energy of ATP. So ATP-driven sodium-potassium pump stores, right? Well, that's related to secondary transport. So I'm going to explain it in a minute. So this is just regular primary transport, right? Now, what is secondary transport? So secondary transport, when we have transport proteins shown right here, they do not directly use ATP. So you see there is no ATP usage in this example, but they depend on a primary transport. So if primary transport is not functioning, if this protein is destroyed, this transport will shut off as well, right? When, for example, for facilitated diffusion over here, right? So over here for facilitated diffusion, both types is a specific channel or non-specific channel or aquaporin. You don't have any active transport involved. Even if you destroy all of this ATP sodium potassium pump, this movement still continues, right? However, in a secondary active transport, if primary pump is destroyed, then it shut off the secondary as well, right? So this is how it works. The ATP-driven sodium-potassium pump stores energy by creating steep concentration gradient for sodium entry into cells. So we pump out sodium, and what we do? We create sodium gradient. So we create high concentration of sodium outside the cell, right? As sodium diffuses back, so sodium diffuses back over here across the membrane through a membrane cut transporter protein. So that's our core transporter protein. It drives glucose against its concentration gradient into the cell. Now we create sodium gradient. Now sodium moves down its concentration gradient. However, glucose moves against the concentration gradient. So here we manage to move glucose from area of low concentration to area of high concentration without direct use of ATP, but we depend on this primary transport. So it's called also co-transport. We have symport or antiport. Sometimes symport means we move in the same directions, two molecules or two substances like ions and molecule, or we can move them in the opposite directions. So that's gonna be antiport. Another examples of active transport are endocytosis and exocytosis. Endocytosis includes phagocytosis, pinocytosis, receptor-mediated endocytosis. So let's look at the example of endocytosis. Now, endocytosis is the packaging of extracellular materials into a vesicle for transport into the cell. Now, receptor-mediated endocytosis. Target molecules bind to receptor proteins on the membrane surface, triggering vesicle formation. So here's our target molecules. Our goal is to bring them inside the cell. But the vesicle not just forming over here. First, those target molecules, they bind to special proteins on the surface of the cell membrane, and they trigger formation of the vesicle, and vesicle moves inside. Vesicles that containing target molecule. So that's our endocytosis, because we move material into a vesicle into the cell, right? Factor affecting rate include number of receptors on the plasma membrane and the concentration of target molecules. And substances involved target molecules called ligands. Now, pinocytosis. In pinocytosis, vesicles form at the plasma membrane and bring fluids and small molecules into the cell. This process is often called cell drinking. All right, so here you have a cell, and we have like small pinocytic vesicle that it's enclosed just a fluid with some dissolved molecules. Factor affecting rate, stimulus and mechanism not understood, right, over here, and substances that are involved, extracellular fluid with dissolved molecules such as nutrients. Oh, and I forgot to give you example. Let's go back over here, receptor-mediated endocytosis. Example, cholesterol and ions are transported this way. example of pinocytosis, once the vesicle is inside the cytoplasm, water and small molecules enter the cell across the vesicle membrane. So example would be movement of water and small molecules that are dissolved in water. Phagocytosis. Vesicles form at the plasma membrane to bring solid particles into the cell. This process is often called cell eating. And factors affecting rate, presence of pathogens and cellular debris, so that's what we're going to bring in, and substances involved, bacteria, viruses, cellular debris, and other foreign materials. Now, in this example, cell forms special cytoplasmic extensions called pseudopods. So pseudopodia is formed that engulfs the particles and put it into the cell. So here we also forming vesicle, but we have this cytoplasmic extension called pseudopods that encircle the particle and bring it in. Right, so those are three examples of endocytosis. And exocytosis, it's a movement of substances inside the vesicle and movement them outside the cell. So vesicle formed from Golgi apparatus, and they move towards the plasma membrane, and then they release their substances that they have inside. So they release it and secrete them out of a cell. So we have two types of exocytosis, continuous secretion or constitutive secretion and regulated secretion. When we have this continuous secretion, then newly synthesized proteins, or it can be, other substances, not necessarily proteins, are just constantly secreted, and there is no regulation. In a regulated secretion, you need a signal or ligand. That's a molecule, maybe will be hormone, for example, that will bind to the receptors on the surface of the cell, and it will trigger the development and movement of vesicles and secretion of product. Okay, and the last part of this chapter is probably the most complicated. It's called the cell membrane potential or resting membrane potential. Now, all cells establish electrical potential called the resting membrane potential, that is from -50 to -100 millivolts. So the average is -70 millivolts. Just remember this number, -70 millivolts is an average. So it's right between 50 and 100, right? Now, to understand the formation of membrane potential, we need to review what electrical potential of voltage is and what electrical current is. So electrical potential of voltage is the separation of opposite electrical charges. So if you have your battery, and your batteries have some energy stored, right? So you use battery to, for example, power your flashlight. So this energy for your flashlight comes from a battery, and battery has this energy stored in the form of voltage. And why we have this voltage? Because we separate positive charges and negative charges. So when you separate charges, then you create potential energy in your battery, right? And when you connect positive and negative ends of the battery, you have movement of these electrical charges that called electrical current. Right now, how it is relative to our topic? Well, because your cells are actually, so here's example of very simple cell. You have extracellular fluid here and intracellular cytoplasm inside, and you have a cell membrane. Now, what cells do, they separate charges across the cell membrane. So your cells are positive outside and negative inside. So here you have separation of charges. You create potential energy, and you can measure this energy using voltmeter. And here's your average, minus 70 millivolts, right? So that means every cell in your body, not just nerve and muscle cells, but any cell in your body creates this resting membrane potential, separate the charges, and store potential energy. Now, how much energy is minus 70 millivolts? Well, first, minus 70 tells you it's negative inside. But how much is that, minus 70 millivolts? So if you look at your regular batteries, AA, AAA, C, and D, they all about 1.5 volts. And if you make a simple calculation, you will see that minus 70 is about 0.1 volt. 0.1 when your batteries have 1.5. So actually, the amount of electricity that our body has is amazing. So resting membrane potential is separation of oppositely charged particles or ions across a cell membrane that creates a potential energy measured as voltage. So resting membrane potential, RMP, it's a voltage measured in resting state in all cells from minus 50 to minus 100 millivolts. And now, big picture. So let's see if we can make sense of this diagram. So what do we have? Well, this is our extracellular fluid, and this is our cytosol that is a part of cytoplasm. And here shown in blue is plasma membrane, right? So in the plasma membrane, you will have different proteins embedded in the plasma membrane. Now, one of these proteins is called sodium-potassium pump that uses ATP. And what it does, it pumps sodium out and potassium in, right? So pretty much every single cell of your body has this sodium-potassium pump that constantly pumping sodium out, potassium in. 3 sodium out, 2 potassium in. Now, why? Why do you need to do that? And actually use a lot of energy to do it? Because what sodium-potassium pump does, it creates chemical gradient for sodium and potassium. Now you have the whole bunch, lots of sodium outside, and lots of potassium inside. Now, mnemonic to remember it, your cell is salty banana because you have salty. Remember, salt is sodium chloride. So you have sodium outside and banana rich in potassium and high concentration of potassium inside, right? So your cells are salty bananas, sodium outside, potassium inside, and this gradient is created by sodium-potassium pump, right? Now, what else do we have? We have other proteins over here called leakage proteins or leak channels, right? So over here, leak channels. Now we have leak channels for potassium and we have leak channel for sodium. Leak channel means they're not regulated, they always open, and they just allow sodium over here, or potassium, move down its concentration gradient. So kind of like there is always some kind of movement here. Now look, that these sodium ion channels, the plasma membrane are 50 to 75 times more permeable to potassium than sodium. So this leak channel is not, we don't have that many of them, right? So for the purpose of this understanding of this lecture, we're gonna ignore this channel at all because we have 50 to 75 times less sodium that moves in than potassium moves out. So just imagine we don't have this. Let me just even kind of like draw the permanent crossover here, right? So remember, we have it, but we're ignoring it because it's not that critical at this moment, right? So now what we have after we ignore this channel, we have lots of sodium outside, lots of potassium inside, and this is chemical gradient, but it's not electrical gradient, because when sodium moves out, positive charges move out. When potassium moves in, positive charges move in. So we don't have separation between positive and negative charges. But what happened, because potassium, now you have high concentration of potassium inside, it starts leaking out, right? That makes sense? Because it leaks down its concentration gradient. And every time when potassium moves out, it takes its positive charge with it. And cells became more and more negative inside, because you have large proteins inside that are negatively charged, right? So instead of being neutral, the cells became negative inside because you constantly loses this potassium. And cells became positive outside because you gain lots of potassium outside, right? And sodium kind of slowly returning back, right? Giving some changes to this resting potential. But again, remember, we decided not to look at the sodium leak channel at this moment, right? Okay, so this is how we establish this electrical gradient, right? So to create chemical gradient, we need sodium potassium pump. And because we created this chemical gradient, potassium now leaks out and we create electrical gradient. And at some point, this movement reaches equilibrium, right? And this equilibrium can be measured, and it will be about -70 millivolt. That's what we call resting membrane potential, right? Now, why would your cells need this resting membrane potential? Because that's energy stored across cell membrane. And now this energy can be used for different cellular processes. Right. All right, let me see. Yeah, so now we'll go over the steps of generation and maintenance of RMP, or resting membrane potential. First, sodium potassium pump continuously ejects sodium from the cell and carries potassium back in. 2, some potassium continually diffuses down its concentration gradient out of the cell through potassium leakage channels. 3, membrane interior becomes negative relative to exterior because of large anions trapped inside the cell. So if you constantly potassium leaking out and you have large proteins that negatively charge inside, then the membrane cell became negative inside. Number 4, electrochemical gradient begins to attract potassium back. Well, that's what I forgot to tell you. So let's go back just for a second. We go back over here, and we're gonna say like, okay, well, when potassium leaking out, right? Now what's gonna happen? Now you have negative charge inside, positive outside. Now negative and positive attract. So some of this potassium will move back in because they attracted to this negatively charged proteins. So some potassium moves out because of chemical gradient, and some potassium moves in because of the electrical gradient, because it's attracted by negatively charged proteins inside. That's what number 4 is. Now we have electrochemical gradient that begins to attract potassium back into cells. Resting membrane potential is established at the point where the electrical gradient balances the potassium concentration gradient. A steady state is maintained because the rate of active transport is equal to and depends on the rate of sodium diffusion into cell. Right, so we have the steady state right? When we balance electrical and concentration gradient for potassium, but remember the sodium, even we ignore it, but sodium still kind of leaks in. But you have a mechanism to take care of it because you have your sodium potassium pump, so you pump it back out. So that's how you create this stable state that is called a resting membrane potential. Okay, so that's it for today's lecture. Thank you very much for watching, and I hope it was helpful. If you have any questions, actually, you can leave your questions in the discussion sections. Just put a comment underneath the video and let me know if this video makes sense for you or not, or you want me to go over other parts of these processes that we discussed, right? But anyway, thank you so much for watching again, and I hope it was helpful.